right, shall we kick it off?
Yeah.
And let me quickly introduce Daan van Aalten. Daan is professor of the Aarhus University in Denmark, and Daan is leading the lab, one of the two major labs, the team who is focused on OGT-XLID, and today Daan is bringing the latest preprint and presentation for the latest preprint of the work his team was leading. Daan, happy to have you here. Thank you for your time. Appreciate it. And please go ahead and take over the.
Okay, that's fine. So I can't see anybody. So if you want to interrupt and ask, which is probably better than asking your questions at the end of the talk, then please do so by just switching on your microphone and asking a question. Yeah. So Andre has asked me to tell you a little bit about a paper that we just published as a preprint, and we can talk a little bit later about what that means, preprint versus a peer-reviewed paper. And so this is the paper, and so the title of that is Pathogenic O-GlcNAc Dyshomeostasis associated with cortical malformations and hyperactivity. And so I've talked a few times to this group before and also presented at the Cure OGT meeting. So I'm not going to introduce what O-GlcNAc is or what OGT-CDG is. If nobody complains about that, then I will just go to the first slide. This is also a slide I've shown before, and it's really it helps our thinking in the lab, and maybe even in the field for other people. And that is at this point, we know that it's missense variants in OGT that are associated with the disease, but we don't know actually at all why that is. And that is because OGT does a lot of different things and is involved in a lot of different processes. And at this point in time, any of these processes individually could go wrong, or they could go wrong in a combination. So the obvious one that I guess has been intuitively the most attractive to pursue initially is the one of hypo-O-GlcNAcylation. So that means that if OGT stops working because of an error in the OGT enzyme, then it will produce less O-GlcNAc on proteins, and O-GlcNAc on proteins is important for development of the brain and for neuronal function. And therefore, that is a likely hypothesis as to how this disease comes about. But there's a couple of other ones, which is that actually we know that in many cases in these variants of OGT, there is actually a change in the expression of the opposing enzyme called OGA or O-GlcNAcase. And we can't exclude that actually it's a change in OGA levels that contributes to this disease. Also, OGT acts as another type of enzyme called a protease, so an enzyme that cleaves other proteins to bits. And one of the, or actually the only protein that it cleaves into parts in the human cell is a protein called HCF1 or host cell factor 1. And this protein is very important for development too. And indeed, variants, so errors in the HCF1 gene, also give rise to intellectual disability. So it's possible that if OGT error, so an OGT variant, leads to loss of activity to cleave this HCF1 protein, that could also explain, at least in part, the symptoms that we see. Then neurons are particularly sensitive to misfolded protein. And actually, you will all know examples of that, like Alzheimer's disease and Parkinson's disease. Those are examples where the folding of proteins in the cells goes haywire to such an extent that they actually form insoluble aggregations in the cell, leading to toxicity to neurons. So the neurons, they can't clear this misfolded protein. We can't exclude the possibility that variants in OGT lead to misfolding of OGT, and that's what causes aggregation and then neuronal toxicity. And finally, another very important function is that we know that the OGT has other functions than just the O-GlcNAc transferase activity, because it interacts with so many different proteins in the cell. And so there is the OGT interactome hypothesis, which means that errors in the OGT protein lead to a slightly different way of folding, and that then means that it loses interactions with so many other proteins that is also important for, but in an O-GlcNAc independent manner. These are sort of, 5 possible mechanisms. But there's also 3 different levels at which these mechanisms could have an effect. And this is also really, really important to help us think about where to focus our research. And one is brain architecture. And with that, I mean that something goes wrong during development of the embryo, so before birth, which causes a structural problem, a defined structural problem in the structure of the brain, and that is then something that is carried on through life, because a brain really only develops during embryogenesis and in the first couple of years after birth. It's also possible that actually brain architecture is normal, but it's a connection between the neurons that have gone haywire. So neurons need to talk to each other to make electrical circuits, essentially like you have in a computer, that are able to help us think and do and coordinate our limbs in the way that we want. And it could be that it's the connection between these neurons that has gone haywire as a result of any of these 5 mechanisms above. And then finally, it's possible that the brain structure is normal, all connections between neurons are there. It's just when they start talking to each other that something is wrong. So this could be like a very small electrical component in a computer that has gone wrong, so that even if neurons are properly connected to each other, the actual transmission of the signal from one neuron to another doesn't work. And this is really important when we think about maybe in the future a treatment of OGT-CDG patients, because in adulthood, if there is a structural brain problem that was formed during embryogenesis, it's very unlikely that this can be reversed, because basically you would want to correct a defect in the brain itself. If it's about neurons forming connections or breaking connections, that's maybe something that we could still do something about. And if it's just to do with the way that neurons talk to each other, actually that is actually very likely that we will be able to do something about it. And so this is actually what this paper is trying to address, or at least one of the questions. Specifically, it's trying to see if there are defects in the brain structure as a result of embryogenesis or the first few months after birth. So as all of you know, we have made 3 mouse lines carrying 3 different OGT-CDG variants, and the one I'm going to talk about today is the C2Y variant, which is found in a Danish family. In a Danish family, the mother is a carrier, so she has a healthy copy and a diseased copy of OGT, and she has passed 1 healthy copy each to each of her 4. Sorry, it says she's passed on 1 copy to each of her 4 sons. In one case, it's the healthy copy, and in the other 3 cases, it is the disease variant. And I think I've shown you this before. This is actually one of the very, very few cases that we know of elderly OGT-CDG patients. So these guys are about 60 years old now. They have intellectual disability, developmental delay, autistic features, seizures, dysmorphic features, some skeletal defects, and also things like anxiety have been observed in them as well. And so when we then use a technology called CRISPR that maybe you have heard about, it's a technology to make a change in the DNA of any organism, in this case a mouse, and we made the same change in the mouse as we saw in this group of human patients. And so what we then do is we make this change, then we have to grow lots and lots of mice, because every mouse is a little bit different from each other. So in order to see an effect, we need very large cohorts, very large groups of mice to make the things that we look at statistically significantly different. And one of the things that we did, and I actually showed this, I think already at the Cure OGT meeting in November last year, is MRI. So this is magnetic resonance imaging. This is a way of looking at the structure of the brain while the brain is still in the skull, and indeed even sometimes in live mice, and of course also in live humans. And so what we see here is that the cortex, which is the outer sort of area of your brain, which is associated with most of our cognitive abilities, is thinner by, in some cases, up to 1/4 millimeter size. And of course, mouse brain is very, very small, so that is a relatively large difference. But with the MRI, we could just see that these brains were slightly smaller. We could not detect any defects in them. And this is really the key of the manuscript that I am discussing here, is that we have done some additional analysis, which is showing the following. So what you're looking at here now is slices of the brains of these mice. So we take very, very thin slices that we can put onto a microscope, and we can use a special stain so that we can see neurons and the other types of cells that we have in the brain. And the top 3 panels are an example of the cortex of wild-type mice. And you can see on the left-hand side at the top in particular, very nicely, this sort of layered structure. So in the cortex, you have different layers of neurons that talk to each other. And these neurons, they all migrate from an origin deep in the brain to form this beautifully layered structure. And this is what it should look like on the top left-hand corner. Basically, nicely layered arrangement of neurons. And so when we look in the mice carrying this OGT-CDG variant, we see a couple of very surprising things. I should say that these defects are at a scale, at a size that is below the detection limit of the MRI. And also, these kind of defects are not always found in exactly the same place in the brain. So if we take 16 mice and we record MRI and we average them out, which is what we would have to do, then we don't see these defects. But when we go and look under the microscope, then this is what we can see. So first of all, an initial worry is that if you take a very thin section of the brain, then the brain is very fragile, and that as a result of sectioning it, you can create defects in it. And that's kind of, if you look at the wild-type mice, and then at the top right-hand panel, you can see one of these little white holes, and that's probably, and you can see all this streaking, these white streaks, that is just as a result of cutting with a knife a very, very thin slice of the brain. But when we look at the mutant, we can see something entirely different. So we can see these sort of structures. They look like holes. They're not holes because they're probably filled with a particular type of subneuronal structure. But we can see these holes, and we can see that around these structures, there are layers of neurons that shouldn't be there. They should just be a properly spaced layering of the brain, as we can see in the normal mouse at the top panel. So basically, you should compare the top left-hand panel in the wild type with top left-hand panel of the C91Y and look at the differences there. So we can see within the cortex that there are these very strange arrangements of neurons that shouldn't be there. And also in the very outer layer, as you can see on the right-hand side, we have these dips. And again, you can see that these dips, the layering of the neurons follows these dips. So this is not a dissection defect. So basically what this paper shows is that in this mouse, for this variant, there are some structural defects in the brain. That is the main message of the paper. Now, there are lots of caveats. So first of all, this is a preprint and not peer-reviewed science. What that means, I shouldn't be saying pre-pint, by the way, preprint. That means that this is something that we in the lab have done to the best of our ability, but now experts in the field anonymously will read this paper and will do their best to try to find shortcomings or maybe even errors in it. And then we will get those comments and we can improve our work, and ultimately then there will be sort of a version of record published in a scientific journal. So it's important that this is basically how we see it, but other people looking at the same data might have questions that we have not thought of. The second one is that a mouse is not a human. The structure of the brain of a mouse, yes, okay, it has the basic components of the structure of any vertebrate brain, but still, for instance, has a huge olfactory bulb, which we don't have, right? A mouse has a very good sense of smell. We don't have that, so we don't have that bulb. The mouse does. And so there are obviously quite a lot of differences between a mouse and a human. The other one to consider is that this is a male mouse. It doesn't necessarily tell us about what happens with OGT-CDG variants in females. Of course, in this case, the patients were also male, and therefore it was appropriate to make a male mouse. But it could be that in female patients, actually, there are different mechanisms that are at work. It's also an adult mouse. So yes, we look at these mice, I believe they're 3 months old. But we don't know whether this was there when these mice were born, or whether it was present when these mice were born to a lesser extent. So there's a lot more work that needs to be done to try to find out when this happens and to what extent. The other thing to keep in mind is that we already know from the 75 different OGT-CDG cases that we are aware of, that there's a huge heterogeneity, a huge variety in symptoms in these patients. And so, okay, this may be happening in this particular variant, but it doesn't mean that therefore it should also happen in all other variants. And it's even more tricky because it could even be that the same variants, so if you imagine a child being born to 2 completely unrelated sets of parents, but by accident having the same error in the OGT gene, they will develop different symptoms too. And that is because the genetic background provided by mom and dad provides a different way of OGT to work and to interact with all the other proteins and genes in the cell. And finally, and this is the most important thing, nowhere in this paper do we claim in any way that this is the be-all and end-all of OGT-CDG. So this does not exclude these non-structural mechanisms that I was talking about before at all. It doesn't even prove in any way, shape, or form that this might be the most important contribution to OGT-CDG mechanism. So just going back to this first slide, it's really important to reflect on that. Is that what this paper says? That yes, okay, in this variant, in this mouse with this genetic background, there are indeed structural defects in the brain, but it doesn't mean that there aren't defects in synaptogenesis and pruning or synaptic transmission, which, as we discussed before, are probably fixable. Or maybe it's possible that these errors in the brain architecture that we uncover in this paper are just a very, very minor component of the cause of OGT-CDG, and the major component lies in synaptogenesis and synaptic transmission. So nevertheless, now that we have this mouse, we can try to do different experiments that are ongoing in the lab right now, which is to do something that you might call gene therapy, which is to try to treat these mice with a virus that carries a normal has been discussed extensively, or glucosamine, to see if that rescues any of the deficits we see. Electrophysiology, so this is where we can actually now measure how neurons in the brain of these mice talk to each other and whether they are properly connected, and then that will tell us about whether that also contributes to the symptoms that we see in these mice and maybe in the patients. We can make other variants, because this is just one family with just one particular variant, and we've discussed many times that there are so many different symptoms for different patients. And then we can use this mouse to try to figure out which of these 5 mechanisms that I showed at the top might be the main player, and then that might give us ideas for developing treatments in the future. And that was the end of my talk, so I will now stop sharing, and I'm very happy to answer any questions.
Well, I guess one question I had was with the screen you pulled up that showed whether it was synaptic transmission or not. So you're saying that depending on the variant, it could be any of those causing the symptoms, but
yeah,
it depends on the variant itself? Okay.
Well, we can't exclude that. I don't know that to be the case, but we can't exclude that. And so it's very important to not think that this mouse now represents everything that is going on in all of the patients that we know about, because we already know that different variants cause different effects. There are some kids that are nearly non-verbal and non-mobile, and then there are kids that have problems with language skills and cognitive behavior, but are motorically completely normal, and so we don't really understand at all why that is. And this is just a first step into showing what might be going wrong. And so if we make 5 different variants that also include some of these TPR variants that we know of, and they all show this particular defect, then that gives a bit more confidence that this is what might be going on in more OGT-CDG patients, but it still doesn't prove that that is what we need to cure, right? So it is possible that what we describe here is a totally irrelevant side effect, but of course it's not normal to see these kinds of defects in the brain.
Okay.
Any other questions?
Yeah, then I think that's a great presentation. Thank you. I'm also curious about the fact that you mentioned that this is just one mouse with one variant, and how shall the research proceed with generalizing the learnings, or is it even possible to generalize the learnings? All variants are different, as what you've shared. What should be the path, and breeding for each variant in mouse is maybe.
So making a mouse for each variant just isn't possible. And I think you've already experienced yourself that you're trying to collaborate with the Jackson labs, who are very, very good at making genetic models of human disease to make the precise variant that your daughter Victoria has, and they are struggling to make the mice. And that's what we've experienced as well. Of course, in your case, your child is a female, And therefore, we don't know that male mice carrying this variant would even survive. So it's possible for females to have very, very, very severe OGT-CDG variants, but actually not be affected at all, because it is the healthy copy that is being expressed, and the disease copy is being inactivated in a process that's called X inactivation. And unfortunately, that works slightly different in mice and in human females, and therefore mice are not necessarily a good model to female mice are not a very good model necessarily to study defects in early development of human females. Yeah.
Oh, Neil, I think you're muted, Neil. Yeah. No.
You answered my first question of why you only used male mice. My second question is from your last slide. You identified, now that you do have these mice created, that you could do other types of testing like glucosamine and other things with it. I just wonder how far along you are with that, and if somebody's spearheading that.
Yeah, so the difficulty with these mice, in order to do these types of studies, you need fairly large numbers, and so you need to breed them, and the breeding of these mice isn't totally trivial, because it's an extinct gene that makes it a bit more complicated. And so and we also don't, I guess, quite know which variants to focus on, because we have 2 other variants in the lab, and we can't do all of these experiments with all of these 3 variants. So we kind of have to make a choice. With these C2I mice, yes, we have an adenovirus associated virus, an AAV that carries the OGT-CDG healthy copy, and we are administering that in mice right now. But of course, the question is, should that be administered, immediately after birth or in adulthood? Because administering immediately after birth, that will maybe tell us whether we can correct these structural defects, but it doesn't tell us whether, when kids are already born and essentially adults, whether then these defects are reversible, which is what, understandably, you will want.
Okay, thanks.
Could they be tested on the flies to start with, and then if there's like a positive outcome, then try it on the mice, or is it just more on the mice than the flies?
I missed the first 10 words of your question.
Sorry. Can it be like tested on flies first?
It could be. So the difficulty with these AAVs is their delivery. So they would have to get essentially. I mean, let's imagine that it's a problem of synaptic transmission. Then, in order to have an effect, they would have to be able to enter most of the neurons in the brain. And so, fly brain is absolutely tiny. The mouse brain is larger, so it doesn't really help. The thing that would help as a first step, which is what we're doing, is to essentially take the brains of these mice when they're just born, and to grow cultures of these neurons and see whether at least these neurons and cultures are willing to take up this virus and make the healthy copy of OGT. Because without that experiment, there's no point in injecting in mouse brains.
Okay. Thank
But
you.
there too, there's a choice, right? Do we focus on gene therapy or viral delivery of healthy OGT? Do we focus on glucosamine, or do we focus on OGA inhibitors? That's kind of like a key choice, and we certainly can't do all 3 on all 3 different OGT-CG mice that we have at the moment. That just takes too much time.
Yeah. Thank you.
I have a question, Dan. It looks like we still don't have a lot of like enough information about the biology of the disease. Is there anything that you think can be done to start in parallel looking at the treatment options? or until we understand the underlying mechanisms for each of those variants, it is too early to even start the experiments?
No, I think starting with treatments is useful, not only from a a trajectory towards the patient. I think there's somebody who's got their microphone on. Maybe it's Neil. I think maybe Neil can switch off. Yeah, that helps. These two things are a little bit connected because actually, let's say hypothetically, you give mice glucosamine, and that completely recovers their motor skills and their cognitive skills, then that means that the mechanism is O-GlcNAc dependent. If you were to give a virus with a completely catalytically inactive version of OGT, so OGT that cannot do any O-GlcNAc acylation, and that would rescue the mouse, then you know that it's the non-catalytic functions of OGT that have absolutely nothing to do with O-GlcNAc at all. It's just O-GlcNAc is just a bystander effect, but it's not actually the mechanism. So these are experiments that we can do. So the treatments, actually, if you design them in a nice way, they can tell you something about where to look for mechanism.
And how far? So, what are your thoughts about starting those experiments on treatments? Are you
you mean on mice or on humans?
On mice, of course, not humans. Yeah.
Yeah. No. We're doing that, but we kind of just have to. We have to decide which mouse to use. We have 3. We have to decide which type of treatment to focus on first. I shared with you, Andre, this paper that came out again, a preprint, on Sunday or on Monday, which basically the argument of that paper is that OGA inhibitors are neurotoxic. And most of you will be familiar with the fact that OGA inhibitors, a lot of work has gone into them as treatments for Alzheimer's and Parkinson's disease. And basically, in some of the trials, they showed that giving Alzheimer's patients these molecules actually made their cognitive decline faster. And according to this paper, that is because actually these OGA inhibitors are toxic to neurons. We have another bit of work out that we are currently expanding, where we actually show that people that have variants in the OGA gene, so the opposite enzyme of OGT, also have intellectual disability. So that means that if you have loss of function of OGA, you also have intellectual disability. Then do you really want to give an OGA inhibitor, which would do more or less the same thing, right? Disable the function of OGA. So that's why I'm a bit more focused on the viral route and maybe the glucosamine route. The glucosamine route is quite nice because, let's say you have any kind. I mean, first of all, if we show in a mice that there is any kind of improvement, either in terms of the brain development that I just showed, or the cognitive or the motor deficits, then that's something that clinicians can prescribe straight away. With the virus, that's an entirely different story because overexpressing, so putting in lots and lots and lots of another copy of OGT in the brain could be a bad idea for a lot of reasons, and also the route to approval for that is long, whereas the glucosamine one is short. And also with glucosamine, if that works, it really implies that it's an O-GlcNAc dependent mechanism, and then we can really start looking for, okay, which O-GlcNAc proteins, or which proteins lose their O-GlcNAc in these OGT-CDG mice, and that will then give us an idea about what sort of pathways in a cell to look at.
Yeah, that makes sense. That makes sense.
Okay.
Any other questions, guys? While we have Daan here. Dan, I had a question about the OGA inhibitor. Looks like in this article, they've tested all three, at least what I like the old that I know, Eli Lilly, Merck, and SC Neurons. So the three molecules. I looked them up, and these are the three that came out there from this paper. And they say that the negative effect is seen, observed among all three of them. Does it mean for us that that path we essentially can just say, like, we don't need to pursue that anymore because it's more harmful?
Yes. So unfortunately, science is never black and white. So it is possible, or it is, let's say, it is not impossible that the therapeutic benefits of an OGA inhibitor in OGT-CDG outweigh the neurotoxicity that it gives, right? So, and it's also possible that maybe that neurotoxicity actually is in particular types of neurons and not on every neuron in the brain. So it's technically possible that in OGA inhibitors, okay, maybe there is a slight cognitive decline as a result of cortical neurotoxicity, but motor coordination is massively improved because it really helps the neurons in the cerebellum, which is the bit of the brain at the back of our head that's important for motor skills, hand-eye coordination, and stuff like that. And so then it could still have a therapeutic benefit, even though it is a neurotoxic. They were also given at a particular concentration, at a particular amount, that, okay, it was neurotoxic, but maybe if you give them for very, very small amounts for a much longer period of time, there is an improvement. So we can't make, excuse the language, Trump-like statements like this, like, yes, it's good, or no, it's not. Science doesn't work like that. Science is about an infinite an infinite number of shades of gray and not black and white.
That makes sense. And can you help us understand, this OGA inhibitors is basically a small molecule, right? It's a compound that can be taken like orally, right? We don't have to inject it via like virus or anything like that.
Yep.
And that's okay, that's confirmed. And the second, it's generally available for the scientific community, for you, basically, because the molecule, you have access, even though it is a proprietary, like of those three companies, but you have access to it, right?
Yeah, we have access to it, and we could even synthesize it ourselves if we can't buy it anywhere. Yeah.
Is there a way you could just also test it on mice, if it rescues any phenotypes that we observe?
Yeah, but that's what we're just talking about, right?
Okay.
So these molecules have been, at least some of them have been administered in the drinking water of these mice. And of course, you could even feed that in drinking water to female mice that are about to get pregnant. And then you can cover the whole period of development of the mouse embryo as well. Or you can give it to the mice just after they're born. And then you can dissect these sort of different stages of life and different effects. But the fact that there are patients that have missense variants, so variants in OGA that disable OGA function and that have intellectual disability, could also be a little bit of a warning sign, just like the neurotoxicity seen by these OGA inhibitors. And it maybe also explains why all those companies that I and you and others in the field have extensively approached to collaborate are not so keen, is because they know something that they don't want to say, basically. Yeah. Because all of the ACE Neuron had a lot of money to do phase 2 clinical trials, and they pulled out. And I tried very, very hard to engage with them, but in the end they didn't want to. Reasons that they didn't want to disclose.
Now, when Braden was on chemo, and it's totally separate from what we're talking about right now, some of the medications they would put him on, they knew were toxic going in. They had some medicines they could give that would help with the toxicity. Is there anything like that for?
No, I mean that we don't. No, that we don't know, but I mean, I think the example that you're giving your friends is in cancer, right? Most of the things that we give cancer patients are very, very, very toxic compounds, and basically they are just ever so slightly more toxic to cancer cells than healthy cells, and so that's how we slowly kill cancer. So of course, OGT-XLID is not cancer, but I'm just giving an example of how things that are toxins, when used at appropriate concentrations, can still have a therapeutic benefit. And so this we don't know.
Okay.
More of a comment than a question, but I thought before starting the glucosamine testing, have you given the thought of maybe interviewing, parents or, patients that are already doing it in real life, and to see what their outcomes are? So you have at least something to, can get what the real world effects of it are with, individuals that are using it.
Yeah. No, that's a very, very good comment. And here is where maybe sometimes science and your personal lives don't align. As a purely thinking as a scientist, what would be great is that if nobody went onto glucosamine and then we put all the kids on glucosamine, or at least we gave them a pill, and one of them would be a placebo, the other one would be glucosamine, and we look at differences in performance. We can't ethically do that with this group of people at all. There's also the real problem that if we look at anecdotal evidence, so let's say one of you gives their kid a dose of glucosamine, and you see a marked improvement in cognitive skills or emotion skills, and you think, it works. The problem is you didn't have a control, so you don't know whether this was just normal development of your child, or if it was linked to glucosamine. We can't separate these two facts from each other scientifically, and that's what we have to do. So we applied, as some of you know, for a grant to do a clinical trial with glucosamine in a limited number of children in a crossover design. So that means that for 3 months you go on, and another 3 months you go off, and you actually don't know as a parent or as a child what is on and off. These are just 2 bottles that are labeled, and then you do exactly what you suggest, Neil, which is to fill in questionnaires to see whether motor skills, cognitive performance is any better. And then with a large enough group of kids and a large enough number of these cycles of crossing over, you might be able to see something. But this is kind of where I run into a barrier, because I'm just a stupid scientist. I'm not a clinician, and so these are things that we have to do to get up a clinician. And as many of you know, we actually try to collaborate with Zach's clinician Stephanie. We wrote a grant to do exactly this, and that grant was unfortunately rejected.
Yeah, and I have a comment on that, Daan, that even if we have a study, it looks like we also have to select the right variants, because not variants are the same in terms of how they affect. And for some, it's increase of O-GlcNation, right, where glucosamine can help. For some, this O-GlcNation is the same. That's what I heard, I think, from you and from
I mean, that depends what model you look at. So in all 3 mice that we have made with 3 very different variants, they all have an increase in O-GlcNAc in the brain, and they all have a decrease of the O-GlcNAcase protein as a response. So that clearly hints to, at least in these 3 different variants, the disruption of the balance between OGA and OGT and O-GlcNAc being disrupted. All 3 mice showed the same in their brain. So the O-GlcNAc hypothesis is at this stage a reasonable one. It doesn't mean that any of these other 4 mechanisms are included, but kind of it says to me that, yeah, this is probably something worth investigating. And then glucosamine is great, because if that works, okay, not only great for you and your kids, but also great from a scientific point of view because it kind of implicitly proves that it is an O-GlcNAc dependent mechanism. I mean, talking to the parents, some of whom I'm seeing here today, anecdotally, I think it'd be fair to say that those taking glucosamine haven't seen an absolutely dramatic improvement in the performance of their kid. And again, even if you did, you don't know whether that would have happened anyway without the glucosamine, because these are still kids that are developing.
Makes sense.
Yeah, I just, in our situation, we saw an increase in motor skills a couple months into glucosamine, nothing really intellectual. And we're about 5 months into it, and that's all gone away. We're not really seeing any effects at all. So
Yeah,
you just don't know. It right now, it's not really doing anything from, our, layman observation of
yeah.
her.
So you felt that that even those improvements in motor skills kind of went away after 5 months. Is that what you're saying?
Yeah, I think it's about month 2 or 3. And it's I don't want to say increased activity levels, for not more jacked up, not scientific, but there was increase in the activity level. I wouldn't say the execution of it, but just a higher activity level. But that's gone away now. So we're not really seeing any effect at this point.
Yeah, yeah. I think, Sarah, you also said something similar, didn't you, if I remember correctly?
We haven't been given that.
Yeah, we haven't been glucosamine. Okay. Well, then it was maybe Andre who said that you felt that there was more activity. Is that right?
Yeah. Victoria was more active physically. Mentally, she has been struggling. Recently, she started to show some signs of reasonable behavior. And also, I just sent you down the video where she goes on a beam, like the mouse that you showed us like last year.
Yep.
Dan showed us the mouse where a mouse would just kind of go on it's not a beam, it's a stick, and then fall.
Steel bar.
Yeah. Bar and fall then. Right, and Victoria did exactly the same. Exactly, like it was. We could when I saw this behavior, I sent it to Dan just to show that this is exactly mouse and Victoria. But just yesterday, I showed a new video where she actually goes by herself on the beam, turns around and comes back, which is insane for her. This is insane, and she shows some signs of improvement. I don't know if it's attributable to glucosamine. Again, we don't have a control group or anything like that, or she's just growing. There's a ton of ABA therapy, ton of like other therapy that we're adding, just hours and hours and hours that we are just giving her. So maybe all of that combined is working, but we can't tell.
Yeah, no, and what one possibility is that actually development is still going on. It's just hugely delayed. So that things that a 5-year-old or the things that a 2-year-old could do, maybe your 5-year-old is just starting to do. But this is kind of where we are, drifting into the clinical realm, where I should really just shut up and not say anything anymore, because I'm not an expert, and also it's very difficult to sort of keep people from giving glucosamine, because it's something that you can buy off the shelf, and the studies that are out there using glucosamine on people and on mice have a very good safety profile. So they might cause a little bit of diarrhea, but that's it. That's my understanding from the literature anyway.
Yeah. Daan, what do you think about other supplements or drugs that the drug repurposing path? Is it still worth pursuing? I know that you've already done it with flies. It looks like there's still room for opportunity.
Yeah, so we've done it in flies, and we've done it in the stem cell model that we published last year, and we've found a compound, and the same compound was independently found. We saw that on a poster at the last Gordon glycobiology meeting, which was like 4 or 5 weeks ago in Italy by the lab of Susan Walker, and that was a statin, lovastatin. We then, when we then tried this molecule again away from the screen, we don't really see any significant change in the levels of O-GlcNAc or OGA or OGT. So I would absolutely not. Yeah, so for us, actually, that screen, we did it, and we felt that we didn't find anything. But it's interesting that the same compound was found by another lab. But the thing is, O-GlcNAc is like a stress response modification. So if you take your cells or your flies and you put them into a high temperature for a couple of minutes, then you will see O-GlcNAc on proteins go up like mad. If you put your cells into, say, high salt concentration to give them what's called an osmotic shock, you will also see higher levels of O-GlcNAc. So O-GlcNAc is just a way for cells to respond to stress. And so maybe with this drug, lovastatin, you stress the cells and O-GlcNAc responds as a result, but it doesn't actually mechanistically mean anything. But we're not sure, but it's not something we're pursuing any further.
Makes sense. Great.
Okay.
No more questions for now.
Just other than, is there anything else that we can be doing to support you, Dan?
Gosh,
What do you need?
what do I need? I can always use more pairs of hands in the lab. So the
Money.
the salary budget is now spent out until 31st of May 2029.
Okay.
And at which stage, this is the way that the science works. Everybody apart from me will lose their job. So hopefully before that, I can get some funding to get this work to continue. But I mean that's I start worrying about that in about 2 years' time. But for now, I think like the grant to collaborate with Stephanie, the clinician that after Sarah's son Zach, that would have been really good, because it would have given us a pair of hands to do that specifically in the mouse, and she does the glucosamine trial in humans. There are all kinds of sources for rare disease funding. I think the thing that you can do, and which you have done, is to inspire early career researchers in our labs, and of course not just my lab, but also as Andre said, the lab of Lance Wells, who is definitely leading on this in the US, and who I know that Andre visited and that Andre found very inspiring. But I'm very sure that the young people in Lance's lab also found it very inspiring. You could urge your clinicians to collaborate with us. I mean, these are very busy people. They don't just look after your kids, but after hundreds of kids with not just OGT-CG, but 100 different syndromes. So they often don't have a lot of time. And then I think something like the Cure OGT meeting that we had in November, I think that was very useful for us. Because we definitely learned a few things there that we hadn't heard before. Like, for instance, a lot of your kids maybe have a high pain threshold. That was not something that was described before at all, and that could immediately translate into a mouse experiment, for instance, right? And we can try to see about pain or heat tolerance in the mouse. Because what we need from these mice is a very strong symptom that matches what we see in the patient. Because the stronger the symptom, the larger the effect, the more chance we have of then trying different treatments and correcting them, right? If the symptom effect is only very small, it means that we have to use very large numbers of mice in order to see, for instance, the partial effect of a virus or glucosamine or OGA inhibitor. So the larger the effect, the more quickly we can progress. So if it turns out that actually, okay, yeah, we did these experiments of the mouse walking on the bar and falling off, or the mouse being in this T maze, right, where it has to make left, right, left decision. But if it turns out that actually, oh yeah, no, with pain we can see a huge difference between these mice, then that is what we will use, even though it is only one of the symptoms that we can see in the patients. The pain is much easier to measure than mice walking off a bar, because that basically means that an experimenter has to sit there and score whether a mouse falls off or not, which is very labor intensive.
Makes
I
sense.
hope that answered your question.
So quite a lot.
Actually, you know what, Dan? I know that you're like a little bit kind of shying away from really asking for funding, but looks like it is time to do this. And if we have a proposal from you on something specific, a specific project, then we could start focusing our efforts on funding certain things. And we have you, we have Lance's lab, and if this helps move faster, then probably that would be a good idea as well. You're saying pair of hands. I don't think we should think not of like in pair of hands like terms, but in terms of the outcomes, right? Like you're testing this certain hypothesis. For example, OGA inhibitors on mice. This is a pretty substantial, very concrete outcome that we can test like right away. Not right away, sorry. Look, it's at some point. And this is a very good goal for us as a family community to look at and to understand. So if you could think of that, maybe let's.
Yeah. No, I can do that. I guess what I I mean, as an example, I said, yeah, we have these 3 different ideas for treatment. We have these 3 different variants, right? That is 3 times 3 is 9 possible combinations. Even if I put all the people in my lab on just that project, we still wouldn't be able to solve that very quickly. And so my hesitation is the time scale. Is that you might think that this is something that you can know in 3 or 6 months, and it just doesn't work. It doesn't work like that. It just takes a long time to do it properly. And but I mean, yeah, it could. I mean, there will definitely many young people who would be interested in a position like that, right, where they are essentially employed by a patient, or they're employed with funding from a patient organization, and they're really working on stuff that is directly related to the patient. I mean, that's what most young people nowadays want. They want to see the immediate relevance of their research. They're not as nerdy as I am, just interested in the basic mechanism as well, for just for science sake and finding out things. But they want to have this connection, and so that is what is on offer here. And so you would be able to attract really good people. Yeah, I mean, we just advertised for an electrophysiologist, so a person who is going to look in these mice at the electrical connections between neurons and whether they talk to each other properly. And we had some really good applicants, and many of them wrote that they read about Cure OGT, and that is what attracted them to this project. So imagine that there is actually, say, a Cure OGT funded PhD student or something like that, right? But a PhD takes 3 years, and so that is. I mean, that doesn't mean that it would take 3 years to get the outcome that you're talking about. But it's still a slow process, and it's just because we can't just use 1 or 2 mice, right? We have to use like 12 normal mice versus 12 mutant mice, and look at the effects. What concentration do you use? How long do you treat them for? When do you start treating? Right? These are all additional variables in addition to what treatment do we use, which variant do we use. So it's not just 9 experiments, it's like 50 experiments.
Yeah. Yeah, that makes sense. Well, let's at least, yeah. Can you think of it, Dan? What could be?
Of course, I mean, I can write a proposal, and yeah, feel free to discuss that with the other researchers in the field. But I think I can understand that it gives it makes you feel like you're actually doing something concretely, and at the same time, the person involved would feel that they're working on something that is really of potential immediate relevance to you. So I think it could be a win-win situation in both directions. I mean, I can come up with a proposal.
Okay, yeah, that would be great. It's not about us feeling better. It's about doing something that lies on a critical path, really.
Yeah, but that's what I feel. I can understand that you are, to some extent, you're kind of sitting there and you're in this situation, and you feel like there's nothing that you can do, and so this is indeed something that you can do. Yeah, I mean, and also something for which I have very little time to go fundraising in the way that you could go fundraising. And obviously, if some of you have contacts with other funding organizations, or with maybe philanthropes who want to use their personal wealth to help families like you, then I'm very happy to present to them as well and explain what it is that we would do and why this is important.
Yeah, actually, this is a very good point. Hearing from you that this can make a difference and this can really accelerate the path to treatment, I think that would be a very important thing. And even I would ask to record you and make it like a short video on that, so that we could, I don't know, post it somewhere, maybe not somewhere. It has to be targeted. If anyone asks for that, or like whenever the decision time comes, there is a very clear evidence that it's not just us asking for something. It really makes a difference. It really helps accelerate the treatment pursuit.
No, I mean, some of the the other CDGs, right, congenital disorders of glycosylation, I think that they are funded by somebody who through their work got very wealthy and was then keen to share this wealth to fund research that would help this group of patients, and people like that are around. Yeah.
Cool. Yeah, would be great if you just put something, just something like a couple of lines. Not we're not here to read grants, right? But really something that you think worth pursuing.
Yeah.
One last thing that I wanted to share with you. Next week, guys, I'm arranging a conversation with, not a conversation, as a presentation from Jonathan and Naomi from Lance's lab. When we were in Lance's lab last week or 2 weeks ago, we saw a presentation about like the variants, how they're different, that not all of them are the same. I think it's important concept. Dan just really stated that there's also like couple like there's a similar but somewhat different angle that Naomi and Jonathan are coming from. So everyone is welcome. I'm planning that for the next community call, which is on 22nd of May. I just want to make sure that they are confirmed. I have a preliminary confirmation. Just want to make sure that they have time, and they will come. So everyone is welcome again to join this community call. It's essentially next week on Thursday. And of course, you and your lab is welcome to join. Like it's the more people. I know that everyone is busy for sure. It's our interest, but again, maybe your lab would like to listen in.
Yep. No, I mean, if I get the invites and they end up in my calendar, I'll be there. I didn't even know that you were doing this, so I just accidentally came across one recording, and if you actually send an invite to my Aarhus University email, I will put it in the diary, because this is a fine time for me in the evening. I'll almost always be available. The other thing I wanted to say, Andre, is that I mean, maybe not everybody here knows that, but there was this O-GlcNAc meeting in Durham, North Carolina, in July, right? And there is some talk about maybe also having sort of a Cure OGT or small meeting associated with that. My understanding is that at the moment, they're not 100% sure whether this meeting will go ahead, because they don't have a lot of applicants for the meeting. And but it would be useful to know that if you are planning to do this, to know when that is, because that will then help with our travel. For us, it would be better if it was during, you know, kind of like just outside the hours of one of the days of the meeting anyway, instead of attaching, you know, half a day before or half a day after the meeting. It's just, you know, 2 or 3 hours during. I mean, even in the evening. I mean, I don't care. You know, after dinner, I'm happy to talk OGT-CDG for sure.
Yeah. So for now, I didn't change my plans. I'm not coming earlier, so like I will just come for this evening. So and fly the next day. So if there is something that we can arrange, I think it would be without me. But again, I don't need to be in the room really for the researchers to talk with each other. As Stephanie and Mike shared that, basically you can carve out like an hour or two during the main event. And I think that would be a great outcome. Again, there will be Stephanie, Lance's lab, you maybe someone from your lab. Mike will be there, and maybe somebody else who we can also invite there to this small meeting. So let me come back to Stephanie and see what time slot could work for her for the schedule.
Yeah, because I think it's important to understand that this meeting is about O-GlcNAc in any kind of realm of the life sciences, right? So it's about diabetes and cancer, and in immune diseases, some of which could, of course, theoretically be relevant for OGT-CDG. But the actual OGT-CDG session is just one afternoon or something like that, where it's me and Lance and probably a couple of other people speaking. So that would be then the logical time to before or after maybe meet with parents, or if the clinicians want to come and attend, that would be fine too. But I think right now we first need to have confirmation the meeting is actually going ahead. So they're saying that they're maybe don't have enough subscriptions or application to the meeting for it to go ahead. So that would be a shame, because it would be me and 4 people that all work on OGT-CDG that will be participating, and there's quite a lot of things that I'm not telling you yet today, which we would reveal at this meeting for the first time, which I think you would be interested in.
That would be awesome. Well, let's hope.
Would there be an online option for that, or is it has to be in person? Because
no, I don't think so.
It has to be in person.
And it's not our meeting. It's a O-GlcNAc meeting that Mike and Stephanie were planning even before we even started to talk about our OGT conference. It's a very different thing from Duke University. And it's just we're trying to capitalize on that as we did with the
I mean it would be very rare that me with a large part of the lab come to the US, right? That it only would happen for this meeting. I mean, the young people in my lab are scared to death that they will be sent to El Salvador or locked up in jail or something like that. And actually are not coming for that reason. I can tell you, somebody is getting married 3 weeks after the meeting, and he doesn't want to come because of this.
Oh wow.
Yes.
Mm-hmm.
I'll go, but I mean, it's I can drive there.
Yes, so that'd be easy for you. It would be easy to even catch up.
We can't drive there, unfortunately.
Sarah can't drive there.
Quite a drive. But, going back to what you said, Dan, about potentially doing like another sort of conference. I know we sort of maybe briefly talked about that. Is that sort of the plan maybe for next year, to do like a dedicated conference to just OGT?
Well, yes, to OGT-CDG, or maybe by that stage it would be, O-GlcNAc related developmental disorders or something like that. It would still be a relatively small meeting, but because there is quite a lot of funding here in Denmark for organizing such conferences, it means that we can pay for people, maybe even for parents, come and attend the meeting. And I think the funding organizations here would love that kind of setup, bringing together basic scientists. And I mean, it would be, no more than 1 or 2 meetings, because we couldn't 1 or 2 days, because we couldn't fill 2 days with everybody that works on OGT-CDG. Or it would just be an opportunity maybe for actually not so much me and Lance to speak, but all the early career people in the lab. And so I think this is something that definitely we could do, and this is actually something that you can help with, because to organize a meeting like that, I guess you would need something like between $50,000 and $100,000 to get the venue, get the food, get the accommodation.
Well, I know someone that works in events. I'm married to him, so
Yes,
I've got connections.
You've got connections. Yeah,
he's not online at the moment, but I'll sign him up.
Yeah, I'm sure he can't just organize a venue in Copenhagen. But there are many of these funding organizations are actually based in Copenhagen, and they have venues that they would give. So we wouldn't need to pay for the venue, but we'd still need to put people up in hotels, give them food, organize their travel, and that kind of stuff is probably best not to let me do, because it would all go wrong.
It's what I used to do. I used to organize travel for people, so I can do things like that.
Well, actually, if that is a strong desire, and you think that this is just kind of a realistic, we can start planning that for the next year, right?
Yes.
We can start discussing the date, looking at other events that are around, just putting together a list of participants, sending out preliminary questionnaires, asking what date works for them, things like that. Because what we put together in November, that was just such a short notice, because we had a venue, all the people together. We didn't have to pay a lot of money. But here, if it's a standalone event, it has to be planned way in advance. And talking with sponsors is going to be another big thing, right? Because some sponsors were like, "Oh no, that's too far. That's too close. We cannot decide that quickly."
But here in Denmark, there are specific grants for organizing such conferences. So both the Lundbeck Foundation, the Carlsberg Foundation, right, who make the beer, and the Novo Nordisk Foundation, they have grants to organize scientific conferences, and I think they are in that sort of ballpark figure of 50 to 100,000, and then we can really organize something nice. Yeah.
Mm-hmm.
Oh no, that's a how. Let's decide that we need to do this, and after that, that's like Sarah. That's
I'll try out, Andre. I'll get Steve on it. He's got connections. He'll be fine. He doesn't mind hand carrying screens and stuff, does he?
Yes, he does.
That's fine. Yeah, we can sort that for sure. Yeah, sounds good. Thank you.
Awesome. Thank you, Dan. This was incredibly helpful as always.
Yeah, and it was nice seeing you too, Eva and Eve. Yes.
Okay. Same. Okay.
Okay. Thank you. Bye-bye.
Okay, thank you. Bye.
Bye. Bye. Bye.